Good day and welcome to today's fascinating journey into the heart of cell biology. Today we're delving into the world of mitochondria and aerobic respiration, a subject that reveals the fundamental essence of energy production within our cells. Mitochondria, also known as the cell's powerhouse, play an important part in supporting life through a complex process known as aerobic respiration. In this lecture, we'll take a look at the complexities of mitochondria, including their structure and function within the cells. We'll also delve into the mysteries of aerobic respiration, learning how it powers our cells, fuels our bodies, and eventually allows us to go out about our daily lives. So, if you're ready to go on a voyage through the microscopic powerhouse that feeds life, let's start with the metabolic marvels, the mitochondria, and aerobic respiration. Before we move forward with the lecture, I'd like to share with you some of the terms that we're using in this lecture. First is anaerobes. They capture and use energy by oxygen independent metabolism like glycolysis and fermentation. When you say aerobes, they use oxygen or they are oxygen dependent to extract more energy from your organic molecules. In eukaryotes, the use of oxygen as a means of energy extraction will take place in a specialized organelle. We call it the mitochondria. And depending on the cell mitochondria, they can have a very different overall structure. You will see here on the photo or figure on your right, the mitochondria has different portions and different comes into different shapes. Sometimes they have bean-shaped organelles, but they can be also be round or thread-like. The size and the number of mitochondria reflect the energy requirements of the cell. The mitochondria can also fuse with one another. We call it the process of the mitochondrial fusion. The mitochondria can also split into two. We call it the mitochondrial fission. You'll see here a time lapse of your mitochondrial fusion and fission after 87 seconds. Mitochondrial fission is induced by a contact within thin tubules from the endoplasmic reticulum. They can encircle and wrap around the mitochondrion like a noose. The ER tubules initiate constriction, which is then completed through the action of soluble proteins that are recruited to the outer surface of the mitochondrion from the cytosol. What you're seeing the photo on your below is the mitochondrial fusion and fission. Mitochondria is often associated with fatty acid containing oil droplets from which they derive or they get the raw materials to be oxidized in order to make the adenosine triphosphates. They are also the sites of synthesis for some substances, including certain amino acids and the heme groups. They play an important role in the uptake and release of calcium ions, which are essential triggers for cell activities. They can also regulate events involved in cell death or apoptosis, which plays an enormous role in the life of all multicellular animals. The outer boundary of your mitochondria includes the outer mitochondrial membrane or the outer boundary, the inner mitochondrial membrane, which were divided into two major domains. The inner boundary membrane domain, which are very rich in proteins and they are responsible for the import of the mitochondrial proteins. The outer domain lies in the interior of the organelle, which is a series of invaginated membranous sheets called the cristae. So this is the structure of your mitochondrion, showing you the photo or the figure on your right. The cristae houses the machinery needed for aerobic respiration and adenosine triphosphate formation. Because of the mitochondrial structure, their membranes have two aqueous compartments. It means it contains water. The matrix, which is found within the interior of the mitochondrion, and the intermembrane space, which are second between the outer and the inner membrane. So you see the photo on your right, or the diagram will show you the structure of your mitochondria. The mitochondrial matrix contains first the ribosomes, which are sites of protein synthesis, and some molecules of circular DNA to manufacture their own RNAs and proteins. Remember that the mitochondria has also a genetic material, which is a relic throughout to be the legacy from a single aerobic bacterium that took up residence in the cytoplasm of a primitive cell, which was ultimately become an ancestor of all eukaryotic cells. This is coming from the endosymbiotic theory, which proposed that the organelles of your eukaryotic cell was coming from aerobic bacterium that lived inside your cell. Moving forward, this is your overview of carbohydrate metabolism in your eukaryotic cells. There are arrows that are indicated in this particular steps, and we will discuss aerobic metabolism in your mitochondria in the next further slides. The first step in oxidative metabolism are carried out in glycolysis. So you will see here that glucose is phosphorylated at the expense of one ATP. They are rearranged structurally to form the fructose phosphate and then phosphorylated again at the expense of a second ATP. Here are the steps and they are numbered on this figure. The two phosphate groups are situated at the two ends. We have here the C1 and C6 of the fructose chain. The 6-carbon biphosphate is split into 3-carbon monophosphates. The 3-carbon aldehyde showing here is oxidized to an acid as the electrons removed from the substrate and are used to reduce the coenzyme NAD+ to NADH. In addition, the C1 acid is phosphorylated to form an acyl phosphate, which has a high phosphate group transfer potential, and they are denoted by the yellow shading. The phosphate group C1 is transferred to ADP, forming ATP by substrate level phosphorylation. 2 ATPs are formed per glucose being oxidized. These reactions are resulting in the rearrangement and the dehydration of substrate to form an enol phosphate at the end of C2 position that has a very high phosphate group transfer potential. Lastly, the phosphate group is transferred to ADP, forming ATP by substrate level phosphorylation, generating a ketone at the C2 position, and there are 2 ATPs which are formed per glucose being oxidized. Glycolysis, remember, produces pyruvate, NADH, and 2 molecules of ATP. Your pyruvate is transported across the inner membrane and the decarboxylated to form the acetyl-CoA, which enters the next stage. Next is your tricarboxylic acid at TCA cycle. Your acetyl-CoA was fed into a stepwise cycle where substrate is being oxidized and its energy is being conserved. The 2-carbon acetyl group from your acetyl-CoA with the 4-carbon oxaloacetate to form the 6-carbon citrate. And during the cycle, we have here 2 carbons which are oxidized to carbon dioxide and regenerating the 4-carbon oxaloacetate needed to continue the cycle. Remember, there are 5 pairs of electrons from hydrogen atoms of the substrate that are used in the ATP production. 4 reactions in the cycle transfer a pair of electrons to NAD+ to form NADH or to FAD+ to form FADH2. These are the figure on your right, the catabolic pathways that generate compounds that are being fed into the tricarboxylic acid cycle. The primary products of the TCA cycle are the FADH2 and your NADH. They contain a relatively high energy electrons. These electrons are fed into the mitochondrial electron transport chain and essential for ATP formation. This is the glycerol phosphate shuttle where the ATP is formed when high energy electrons are passed from FADH2 through a series of electron carriers that make up the electron transport chain. These are controlled movement of protons back across the membrane. So seeing here the reduced substrates and passing through the membrane, and hydrogen protons on your right, which has they measured here the electron energy from in volts. Okay, so the coupling of hydrogen ions translocation to ATP synthesis is what we call as your chemiosmosis. There are three molecules of adenosine triphosphate which are formed from each pair of electrons donated by your NADH. Two molecules of ATP are formed from each pair of electrons which are donated coming from your FADH2. Again, this is your summary of the process of oxidative phosphorylation. or copper. Next will be the ubiquinone coenzyme Q, which is a lipid-soluble molecule made up from the 5-carbon isoprenoid units. Again, you can see the figures and the structures on this figure. We have also iron-sulfur proteins. Iron atoms are not located within a heme group, but instead they are linked to an inorganic sulfide ions as a part of the iron-sulfur center. The most common centers contain either 2 or 4 atoms of iron and sulfur. They are designated as your or the ferredoxins. These are linked to the protein at the cysteine residues. The carriers are arranged in an order of increasingly positive redox potential. Remember that electrons lose energy as they move along downhill along the chain. Each carrier is reduced by the preceding carrier, then they are being oxidized by the carrier following it. The final electron acceptor is your oxygen molecule, which is being reduced to water. Again, this is the arrangement of several carriers in your electron transport chain. The specific sequence of carriers that constitute the electron transport chain was being worked out using the inhibitors that block the electron transport at a specific sites along the route. And this is very prominent in this experimental use of inhibitors to determine the sequence of carriers in your electron transport chain. So you have here the point of inhibition and the rotinone block in this experiment is being conducted to make use of the inhibitors. Okay, so they will determine how much carriers or what are the myriad of carrier sequences are involved in the ETC. The tendency for the electrons to be transferred from one carrier to the next depends on the potential difference, basically between the two redox centers. And electrons are probably flowing through a special tunneling pathways which consists of a series of covalent and hydrogen bonds that stretch across the parts of several amino acid residues, showing you the figure of an electron tunneling pathways for the yeast cytochrome C peroxidase complex. That figure is the electron tunneling pathways. Now we move forward with the electron transport chain of the inner mitochondrial membrane. Electron carriers can be isolated as part of 4 distinct asymmetric membrane spanning complexes. We have complexes 1, 2, 3, and 4. Once within one of the multi-protein complexes, electrons travel between adjacent redox centers and their relative positions are fixed in place. There are 3 places where your electron transfer is accompanied by a major release of this free energy. The free energy is released as electrons are being passed. They are being conserved by the translocation of protons from the matrix across the inner membrane into the intermembrane space. These protein complexes are also called or described as the proton pumps. Hydrogen ions can hop through the channel by exchanging themselves with other protons that are present along the pathway. These complexes can be added to artificial lipid vehicles, and when given an appropriate electron donor, they are being capable of accepting electrons and use this released energy to pump protons across the vesicle membrane. So an example on your right is the cytochrome oxidase, which is a proton pump found in your synthetic liposomes. The liposomes and their function will be discussed in later. Complex 1 is catalyzing the transfer of a pair of electrons from NADH to ubiquinone to form the ubiquinol, the hydrophilic domain that projects into the matrix. Okay, the hydrophobic portion of the complex are being embedded in the membrane, and where these portions of the complex carry out electron transfer and proton translocation. This is a crystal structure of a bacterial form of complex 1. Next, it will be complex 2. They're consisting of 2 hydrophobic subunits which anchors the protein in the membrane, and two hydrophilic subunits that comprise the succinate dehydrogenase. It gives us a pathway for feeding lower energy electrons from succinate to FAD to ubiquinone. The fat from FADH2 to ubiquinone, which takes the electrons through the three iron sulfur clusters. Complex 2 contains a heme group, and it's being thought to attract a lot of escape electrons to prevent the formation of your destructive superoxide radicals. Electron transfer through this complex 2 is not accompanied by proton translocation. Now your complex 3 is catalyzing the transfer of electrons from ubiquinol to cytochrome C. 4 protons are translocated across the membrane for every pair of electron being transferred. They are released into two different steps. These are protons. Okay, 2 protons are derived from a molecule of ubiquinol which have entered the complex. 2 protons are being removed from the matrix and translocated as a part of the second molecule of ubiquinol. The last step of the ETC is the transfer of electrons from reduced cytochrome C to oxygen. Oxygen reduction is catalyzed by the complex 4, which is a huge assembly of subunits known as a cytochrome oxidase, which is a redox-driven proton pump. For every molecule of oxygen reduced, there are 8 protons which are being taken up from the matrix. 4 are consumed to form the 2 molecules of water, and 4 are translocated to their intermembrane space. Now we move forward with the carriers that transfer the single electrons to reduce oxygen to 2 molecules of water that are very efficient. So these are releasing the partially reduced oxygen species that could damage virtually every molecule in your cell. So this is an example of a summary of your cytochrome oxidase activity. Now we move forward with the engineering linkage and how do we measure blood oxygen. Animal cells and tissues need a constant supply of oxygen to support your electron transport chain during respiration. And we have a lot of ways to measure oxygen in your blood. First is your Clark electrode, which measures the voltage across the metal contacts, which a current that is created by a set of oxidation reduction reactions involving oxygen. And next, which is the most common, which is the pulse oximeter, measuring the blood oxygen inside the patient continuously using a color measurement. So you see a photo of a red and infrared light and a photo detector. There are 2 components of your proton gradient. Moving forward, this is a concentration gradient between matrix and the intermembrane space, which creates the pH gradient, or we call it the delta pH. The separation of charge across the membrane potential creates an electric potential in psi. Energy present in both components of the gradient is called the proton motive force, or the delta P. So you see here a microscopic visualization of the proton motive forces in your mitochondria. And experiments are showing us that the ionic gradient can be used to drive a reaction in which the ADP is being phosphorylated to ATP. The driving force of this proton motive force is being established by the electron transport. You see here the figure on your top. The top figure is the machinery for ATP synthesis. The second is the ATP formation in membrane vesicles, which are reconstituted with the sodium and potassium ATPase in the exchange. Okay, so we move forward with the structure of the ATP synthase, which is an enzyme. Look at the F1 particle, which is the catalytic subunit and contains 3 catalytic sites for ATP synthesis. The F0 particle attaches to the F1 and is being embedded in the inner membrane. Your base contains a channel through which the protons are conducted from the intermembrane space to the matrix. Well, the basis of ATP formation according to the binding change mechanism, remember the bind changing mechanism states the following. Energy is being released by the movement of protons, which is not used to drive the ADP phosphorylation directly, But principally, these are changing the binding affinity of the active site for the ATP product. Each of your active sites progresses successively through three distinct conformations which have different affinities for substrates and product. ATP is being synthesized by a rotational catalysis in which one part of the ATP synthase rotates relative to other part. And there's a lot of evidence, one of which is the structural basis of the catalytic site formation. Okay? And the structure of each of the catalytic sites in a static enzyme differ in their affinity for your nucleotides. We have here the L, the T, and the O conformations which are found in the three beta subunits. The gamma subunit of the enzyme is perfectly positioned to transmit conformational changes from the F0 membrane sector to the F1 catalytic sites. And showing you the figure of the binding chain mechanism for the ATP synthesis. The gamma subunit end is asymmetric, which is its different phases interact with the different beta subunits, which causes them to adopt different conformations. And during a single catalytic cycle, it rotates a full 360 degrees, causing each of these catalytic sites to pass sequentially through the L, T, and O conformations. Then condensation of the ADP and your phosphate group to form ATP happens at the T conformation. So this is again showing you different steps of the binding change mechanism for ATP synthesis. Evidence for the rotation of the gamma subunit have been shown that there is a genetically engineered version of the F1 portion of the ATP synthase was made, and fluorescently labeled actin filaments was attached to the end of the gamma subunit. When incubated with ATP, the fluorescently labeled actin filament was seen to rotate like a microscopic propeller. This is a direct observation, figure on your right, of a rotational catalysis, schematic, and their video frames. Using the proton gradient to drive the catalytic machinery, we can magnify the role of the F0 portion of your ATP synthase. The path taken by protons as they move through the F0 complex, and how does this movement lead to the synthesis of ATP. The C subunits of the F0 base are being assembled into a ring, which you seen earlier, that resides within the lipid bilayer. The C ring is physically bound to the gamma subunit of the stalk. The downhill movement of protons through the membrane drives the rotation of the ring of your C subunits. And the rotation of the C ring of the F0 provides a twisting force which is powered by torque that drives the rotation of the attached gamma subunits, leading to the synthesis and release of ATP by catalytic subunits of the F1 ring. So this is a model of the proton diffusion coupled to the rotation of the C ring in the F0 complex. C ring subunits move past a stationary subunit, alpha subunits. Okay? Protons are picked up from the intermembrane space by a C subunit and carried around a circle before they are being released into the matrix. The proton binding residue of each of the C subunit acts like a revolving proton carrier. Movement is being driven by the conformational changes associated with the sequential protonation and deprotonation of the acidic residue of each of the C subunit. Remember that the association and dissociation of 4 protons would move the ring 120 degrees Celsius and lead to the release of an ATP by the F1 complex. So you will see that the movement here of the model is powered by the rotation of the C ring, again in the F0 complex. Mitochondria are engaged in other processes and that rely heavy on the proton motive force for energy. This force drives the uptake of adenosine diphosphate and the phosphate group into the mitochondria in exchange for ATP and hydrogen ions respectively. The proton motive force may be used to pull the calcium ions into the mitochondrion, which will drive the events of what we call as the mitochondrial fusion. So these are the summary of the major activities during the aerobic respiration in the mitochondrion. Moving forward, we have the peroxisomes. Peroxisomes are membrane-bound vesicles that contain oxidative enzymes. They function to first is to oxidize a very long-chain fatty acids and synthesize plasmalogens, a class of phospholipids. They also import preformed proteins which are engaged in oxidative metabolism. Hydrogen peroxides, a reactive and toxic compound, is formed in your peroxisomes and is being broken down by the enzyme catalase. So there are a lot of information about peroxisomes which are really of a clinical importance. And peroxisomal diseases are a type of medical diseases that affects the nervous system as well as many other organ systems. Example is your X-linked adrenoleukodystrophy and peroxisome biogenesis abnormalities have been shown as the two typical examples. Thank you so much for watching the Metabolic Marvels, the mitochondria, and the aerobic respiration. I hope you've learned something today, and stay tuned for the next lecture for cell biology. Have a nice day and stay safe. Goodbye.